Right. So KREBS cycle, it is also known as TCA cycle. Switch. TCA cycle. Right. So KREBS cycle, it is otherwise called as TCA cycle, tricarboxylic acid cycle, citric acid cycle, which we have already discussed. Right. So it occurs in mitochondrial matrix. Right. So it was discovered by Sir Hans Krebs in 1937 in Asian breast models. So for which he was awarded Nobel Prize in the field of medicine. Right. So most of the energy remained in the pyruvate is oxidized in the Krebs cycle. Okay, and it is also degraded. Pyruvate enters into matrix of mitochondria by pyruvate H plus importer, which is a carrier protein actually, and oxidatively decarboxylated into acetyl-CoA. So this is important compound in Krebs cycle. So Krebs cycle is started with acetyl-CoA, and it is regarded as connecting link between glycolysis and electron transport chain. So this conversion of pyruvate to acetyl-CoA is, it is catalyzed by a multi-enzyme complex, which is known as pyruvate PDH complex or pyruvate dehydrogenase complex. So which contains different functions. Okay, so the CO2 or carboxyl of pyruvate is first removed as CO2. Okay, so this is the first decarboxylation reaction in the cellular respiration. The pyruvate fragment is then oxidized to acetate. Okay, and 2 electrons released in this reaction are transferred to NAD+. Actually, so to be precise, the electrons are received by FAD. From FAD to FAD, those 2 electrons are immediately transferred to NAD. Okay, so we generally write it as NAD. Okay, simply shortcut. So then for this acetate, which is formed here, okay, that is later added or combined with coenzyme A. So it is so this coenzyme A is actually formed from vitamin B5 pantothenic acid, sulfur containing substance. So combines with acetate to form acetyl-CoA. So which is prerequisite for the citric acid cycle to be continued. Okay, then this is how the reaction takes place. This is pyruvate, the first decarboxylated, then it is entered. These 3 steps are combinedly catalyzed by PDH complex. Okay, so here it requires thiamine pyrophosphate, okay, and lipomide, what we call it, a B vitamin derivative, okay, and Mg+2 ions, NAD+ ions, NAD+, okay. So with the help of all these, the acetate which is formed here is combined with coenzyme A to form acetyl-CoA. So this acetyl-CoA, it can freely move within the matrix and it can go outside also freely, right? So that means the both membranes of this mitochondria are permeable to acetyl-CoA. So these are the 10 reactions which are, part of citric acid cycle. Citric acid cycle is started by the condensation of acetyl-CoA with oxaloacetate. Okay, so anyway, about 65 to 75% of ATP is synthesized in KREBS cycle. It involves the oxidation of acetyl-CoA to CO2 and H2O. It is final common oxidation pathway for carbohydrates, fats, and amino acids. So one thing we should remember that just like glycolysis, many compounds, many intermediates, okay, so they enter into the citric acid cycle as an intermediate compounds, and some compounds even they may leave the citric acid for the synthesis of various different amino acids, fatty acids, triglycerides, phospholipids. Okay, so like that. So there are many intermediates, they can enter directly and they can leave at any stage. Okay, so for the synthesis of various other substances, right? So reactions of citric acid cycle. See, the first reaction is formation of citrate. Just now we have discussed condensation of acetyl-CoA and oxaloacetate by citrate synthase. Okay, here how should we, generally the in the examinations the name of the enzymes are not asked, but some if we follow some tricks, okay, we can remember the name of the enzymes easily. Anyway, here what is forming? Citric acid is forming. Okay, so simply citrate synthase. Synthase means it synthesizes. Okay, citrate synthase catalyze this reaction, and we have discussed it in the previous class. Citrate inhibits PFK enzyme. Okay, it is one of the inhibitors, just like ATP. Whenever sufficient amount of ATP is available, there is no need to proceeding of citric acid cycle and electron transport chain. So this citric acid, it goes to glycolytic pathway, as from the mitochondria to cytosol it goes, and it it binds with the allosteric site of the phosphofructokinase enzyme, which is a protein. So then it deactivates that. So you remember this. And second and third reactions, actually there are in this cycle also there are 10 reactions. Okay, second and third reactions are combinedly occurred. Anyway, citric acid is isomerized to isocitric acid. Okay, so here actual reaction is isomerization of citric acid into isocitric acid. It occurs in 2 steps. So that's why 2 and 3. First, one hydration takes place. Okay, one water molecule is removed, as a dehydration takes place, removed from citric acid to form cis-echoritic acid. Cis-echoritic acid. Okay, so then one water molecule is added to form isocitric acid, which is an isomer of citric acid. Okay, so after this, the same enzyme that is catalyzing, this reaction, echoritase. Okay, so echoritase catalyzes both the reactions. Okay, the removal of H2O and addition of H2O molecule. Right. So here one water molecule is added to form isocitric acid, which is an isomer of citric acid. Both the reactions are catalyzed by voltage. Right. So isocitric acid is formed here, right? So then next reaction also it occurs in two steps. An intermediate compound is formed. So that's why fourth and fifth reactions we call it as, okay. So in this reaction, one carbon dioxide molecule is first removed, okay, from isocitric acid to form oxalosuccinic acid, which is an intermediate compound. So this is first step. Then the enzyme isocitrate dehydrogenase enzyme converts or oxidizes this oxalosuccinic acid, which is, you remember this compound. So it is intermediate compound of many other pathways also. So particularly from amino acid synthesis or their degradation, and the fat synthesis and their degradation. So this is one of the intermediates. So this compound, which is formed in other pathways also, can directly enter into citric acid. So that's why we consider the citric acid is a central. It's like a trophic, island area, like cycle. So in which many compounds they enter, okay, they participate up to some reactions, and they can leave or they can continue according to the availability and availability of ATP and availability of ADP, all those things. Okay, anyway, so this oxalosuccinic acid immediately oxidizes to alpha ketoglutarate acid. So here also this reaction is just like the citrate synthase, like sorry, pyruvate dehydrogenase, like enzyme. This is the reaction also similar. Isocitrate dehydrogenase is a complex, so that catalyzes both carboxylation and dehydrogenation or oxidation. Right. So anyway, alpha ketoglutarate is formed. You can call it as alpha ketoglutarate or alpha ketoglutaric acid. So both are same. Okay. So if H is there, acid. If H is not there, aid. That is the difference. Okay. In this reaction, one NADG, one NADH is formed. Okay. The two electrons, two hydrogens are released from the oxalosuccinic acid. Okay. They are transferred to NAD plus. So to form one NADH. This is so first one in the citric acid cycle for the formation of NADH, and then formation of alpha ketoglutarate to succinyl-CoA. Here also you remember sequentially two NADH molecules are formed, two CO2 are released, one reaction after another one. So it occurs through oxidative decarboxylation catalyzed by alpha ketoglutarate dehydrogenase. This is also so multi-enzyme complex. So here it requires TPP, say just like pyruvate dehydrogenase, thiamine pyrophosphate, lipomide, NAD, coenzyme A. All these are required for this reaction to occur. So in this reaction, CO2 is liberated and NADH is formed. So let's see the cycle. Isocitrate is converted into alpha ketoglutarate. Okay, so here one NADH is formed. CO2 is liberated. The same way, alpha ketoglutarate alpha ketoglutarate is, converted into succinyl CoA. Here, CoA is participating. Okay, this CO2 is removed. Okay, so and another NADH is being formed. మనకు ఇక్కడ మీరేం గుర్తుంచుకోవాలి, నేనేం చెప్పదలుచుకున్నాను అంటే isocitrate and between isocitrate and alpha ketoglutarate, one CO2 is lost, one NADH is lost. In the same way, immediately alpha ketoglutarate to succinyl CoA మధ్యలో, so one CO2 is lost, one NADH is formed. And you remember, మీకు ఈజీగా గుర్తుంటుంది ఇది ఇప్పుడు, how many NADH are formed in Krebs cycle అనగానే మనకి ఈ డయాగ్రామ్ గుర్తు రావాలి. ఏంటిది? 3 NADH. ఓకే, సో దట్స్ క్లియర్, రైట్? మనం మర్చిపోలే, రైట్? సో హియర్ yeah, so succinyl CoA, as here coenzyme thionyl CoA is participating, so succinyl CoA is formed, which is an energy rich compound. So here from succinyl CoA, succinic acid or succinate, so it is also an intermediate compound for many amino acid synthesis and degradation. Succinyl CoA is also one of the intermediate compounds for many fatty acid degradation and their synthesis. So this coenzyme A is very, very frequently that participates in fatty acid metabolisms. So these are the intermediates of other amino acids and fat metabolism. So sometimes succinyl CoA directly can enter into citric acid. Okay, anyway, succinyl CoA is converted to succinate, okay, by the enzyme succinate kinase. Okay, here kinase means always there is an involvement of phosphorus or phosphate group. Okay, so it is coupled with phosphorylation of GDP to GTP. So it is a a kind of substrate level phosphorylation. So the energy which is released in the succinyl CoA removal or coenzyme removal, okay, so that energy is utilized to add one inorganic phosphate to GDP to produce GTP. Okay, and this GTP is immediately converted into ATP by nucleoside diphosphate kinase enzyme. Here, this yeah, so first 2 NADH are formed. Immediately after that, one GTP is formed. Okay, you remember that. And then conversion of succinate to fumarate. Yeah, so succinate is oxidized to fumarate by succinate dehydrogenase, SDH. So it is very famous enzyme. In this reaction, FADH2 is formed. So in that way, it is also important. See, right from the isocitrate, ఇక్కడ నుంచి మొదలుకుంటే వరుసగా 2 NADH2 ఫామ్ అవుతాయి, 2 CO2 అదే రియాక్షన్ లో అవి రిలీజ్ అవుతాయి, అండ్ ఇమీడియట్ గా ఒక GTP ఆర్ ATP అనేది ఫామ్ అవుతుంది. ఓకే, దాని తర్వాత ఇమీడియట్ గా మళ్ళీ FADH2 దేని మధ్య, ఓకే, S and F. Okay, ఇక్కడ ఫ్యూమరేట్ ఉంది కదా, అట్లా గుర్తుంచుకోండి. FADH2 between S and F, FADH2 is formed, right? So why here FADH2 formed? Why not NAD plus? Here the energy which is released is not sufficient, or the electrons which are released here, they don't have the sufficient energy to be coupled with NAD plus. So that's why there is no option. FADH2 is formed, right? So in that way, from succinate or succinic acid, fumaric acid is formed or fumarate is formed. Okay, in this reaction, the enzyme is succinate dehydrogenase, and one FADH2 is produced to form fumaric acid. And immediately next step is hydration, a kind of isomerization. See here the double bond is uh, removed. Okay, so the enzyme participating or catalyzing this reaction is fumarase. Okay, addition of this water molecule. Okay, so it removes this double bond between second and third carbon, and at second carbon, the hydroxyl group, one hydrogen is added to third carbon, one OH is added to second carbon. Okay, so to form a malic acid, which is a dicarboxylic acid. And the last reaction here, so malate dehydrogenase, okay, that converts this, or that takes out the electrons and hydrogens from malic acid. Okay, two electrons and two protons are given to NAD plus to form another NAD, which is third one. Remember, okay. So here, number wise, this is one, this is second, and yeah, second, third, fourth, and fifth. One, sixth. Okay, you can remember as 4 and 5, 6 and 10th. Okay, in these three reactions, NADH is produced. Okay, so malate dehydrogenase catalyzes this reaction to form oxaloacetate. Okay, so here we already know that the oxaloacetate which is formed here, it again ready to combine with another new acetyl CoA. So what we have to understand, what is the theme here? So there is no change in the oxaloacetic acid. Only this compound, ఇక్కడ రెడ్ కలర్ లో ఏదైతే మీకు హైలైట్ చేయబడిందో acetyl group. Okay, so acetyl group is completely oxidized here. Okay, the two carbon containing acetate is oxidized. Okay, there is nothing is happened to oxaloacetate. So what is the actually what happens in the respiration? Oxidation of foodstuffs like glucose. Okay, so in the in the glycolysis, okay, so some part of glucose is oxidized, okay, to form two pyruvates. Okay, so then the pyruvate is oxidatively decarboxylated to form acetyl CoA there in the middle reaction. Okay, so then one CO2 is lost there out of three carbon compound, the carbon atoms, and here remaining two carbon atoms are so lost in the form of CO2, one here, one here. So with this, the what we call the oxidation of pyruvate is completed with the Krebs cycle. Okay, so then okay, yeah. So this is interesting point. Is it a required or is it essential of oxygen for the continuation of PCA cycle? Okay, so why it is not occurring in anaerobic condition? So oxygen, we haven't seen the direct participation of oxygen in the Krebs cycle or in the glycolysis. Okay, glycolysis can occur in the aerobic and anaerobic conditions. Okay, but is Krebs cycle can be continued without the presence of oxygen? No, it cannot be. continued without the presence of oxygen. How? Why? Okay, we have seen here the formation of, the electrons and protons are converted, they are added to, these NAD plus and FAD plus enzymes, molecules. And here the phosphate is added to GDP to form GTP. Okay. And here, particularly these NADH and FADH, and one more NADH in the formation of acetyl-CoA. These are immediately entered into electron transport chain, and they are, oxidized to their native or original NAD plus and FAD plus to form ATP. Okay, so if at all they don't go to electron transport chain, manchi vinandi, avi ganaka electron transport chain lo participate kaakapothe. Okay, so and the electron transport chain is occurred, oxidative phosphorylation is occurred in the presence of oxygen only, oxygen molecule only. Okay, if there is no oxygen, so these will not be converted into ATP. So they will be accumulated, and these will, what we call the accumulated NADH and FADH, they will go inhibit these enzymes. So the Krebs cycle will not be continued. And if at all there are no free NAD plus and FAD plus, okay, also there is no continuation of glycolysis, there is no particularly Krebs cycle, not glycolysis. Krebs cycle will not be continued because the acetyl-CoA needs NAD plus. Here 2 NAD plus are needed, here FAD needed, and here NAD plus needed. For example, if there are no NAD plus, no NAD, there is no Krebs cycle. Then how NAD and FAD are formed? Only in the presence of oxygen in the electron transport chain. There, the NADH and FADH, they are oxidized to NAD plus and FAD plus. So, even though the oxygen is not participating in the Krebs cycle, okay, even though it is not participating in the Krebs cycle, but indirectly it is required for the continuation of next cycle of, next cycle of this Krebs cycle. Okay, so otherwise there will be no NAD, there will be no FADH, right? So there is no direct participation of oxygen in this cycle, but the cycle operates only under aerobic conditions, that is the presence of oxygen, because NAD and FAD are required for the cycle. So can be regenerated in respiratory chain only in the presence of oxygen. Okay. So oxygen యొక్క presence లో మాత్రమే NAD and FAD are regenerated. Okay, so this cycle is strictly aerobic. Krebs cycle, even though there is no direct participation of oxygen, it indirectly depends on oxygen. So it is strictly aerobic. Okay, sometimes set or net exams లో ఇలాంటి questions అడుగుతారు, right? So energetics of TCA cycle. So in this in this Krebs cycle, for a single acetyl-CoA, 3 NADH, 1 FADH, 1 GTP are formed. Okay. In electron transport chain, 1 NADH produces 3 ATPs. So 3 into 3, 9 ATPs. 1 FADH2 can produce 2 ATPs. Okay. So, and 1 GTP is converted into 1 ATP. So 9 plus 2, 11, and 1 GTP. Total 12 ATPs are formed purely only in Krebs cycle. So for one acetyl CoA, for single acetyl CoA. Okay. So for one glucose, only in Krebs cycle 24 ATP after completion of electron transport chain. Okay, and conversion of pyruvic acid to acetyl CoA, if it is added, okay, for one glucose, okay, so there are two NADH2 we should add, so that 6 ATPs. That means if we add formation of acetyl CoA, 36 ATPs are produced in Krebs cycle. Or without acetyl CoA formation, 24 ATPs are produced per glucose. Okay, and if we strictly follow single acetyl CoA, ఒకటే acetyl CoA ద్వారా జరిగే Krebs cycle లో మనకు 12 ATPs అనేటువంటి ప్రొడ్యూస్ అవుతాయి, రైట్? సో దెన్ రెగ్యులేషన్, హౌ ద ది రెగ్యులేషన్ లాంటి ఇది జనరల్లీ జేఎల్ ఎగ్జామ్ లో అడగరమ్మా, బట్ we should have the knowledge for the set or net exams, right? So according to cellular demand of ATP, so sorry, it is very crucial the regulation of citric acid. Okay, so if ATP is demanded by the cell or required by the cell, the citric acid will cycle will be continued. If there are plenty ample amount of ATP, okay, so the citric acid cycle should be stopped. Okay, so for unnecessary energy is lost, so it is regulated by either by enzymes or by levels of ADP. If there are more ADPs are there, what does it mean? It means that the ATP levels are less. Okay, so anyway, the citric acid cycle is regulated either by enzymes or levels of ADP or ATP also. Okay, it may be positively or negatively regulated, right? So first enzyme is citrate synthase, right? So it is generally inhibited by ATP, NADH, acetyl CoA, and succinyl CoA. Why? Because if there are plenty of ATPs or NADH or acetyl CoA or succinyl CoA, okay, so what does it mean? So there are plenty or sufficient amount of ATP is there. That's why they are accumulated. So all these will go to citric acid synthase and they will inhibit either of these. వీటిలో ఏది ఒకటి ఎక్కువ ఉన్నా సో అవి వెళ్ళి సిట్రేట్ సింథేస్ ని అవి ఇన్హిబిట్ చేస్తాయి, ఎందుకంటే మిగతా అన్ని కూడా సిట్రిక్ యాసిడ్ ఫామ్ అయినాక ఫామ్ అయ్యేది కాబట్టి అవి సిట్రేట్ సింథేస్ లేకుండా అవి కంటిన్యూ అవుతాయి. దే విల్ జాయిన్ ఇన్ టు ఇన్ electron transport chain. So if at all these are in sufficient number, okay, if at all these are in sufficient number, okay, so there is the citrate synthase is stopped by these products by product inhibition method, and then IDH, isocitrate dehydrogenase. Okay, so here, for example, in the cell, if the cell is lack of ATP, that means many ADPs are there in the cell. So this ADP will go to isocitrate dehydrogenase and it activates. Okay, and just like in the ATP, NADH అనేవి సిట్రేట్ సింథేస్ నే కాదు, ఐసోసిట్రేట్ డిహైడ్రోజనేస్ కూడా ఇనిబిట్ చేస్తాయి. స్టాప్ దీస్ ఎంజైమ్స్. ఎందుకంటే అవి ఉన్నాయంటే మనకు కావాల్సినంత ఎనర్జీ ఉంది అని అర్థం. కాబట్టి ఏడిపి యాక్టివేట్స్ ఐడిహెచ్ అండ్ రిమైనింగ్ టు దే బ్లాక్ ది ఐడిహెచ్. ఓకే, దెన్ alpha ketoglutarate dehydrogenase. So it is also inhibited by succinyl-CoA NADH. Okay, ఇవి రెండు ఉన్నాయి అంటే so there is no need to continuation of the Krebs cycle. So these two also allosterically block or regulate the alpha ketoglutarate dehydrogenase. And then same we have already discussed here, availability of ADP. Okay, so it is very important for the citric acid cycle to proceed due to oxidation of NADH, FADH2 in electron transport chain is occurred only if ADP is available. But మనం ఏం గుర్తుంచుకోవాలంటే ఈ this is called తెలుగులో ముడి పదార్థం అంటారు, raw material. Okay, if this raw material, that means so the inactive ATP or simply ADP is available, then only the oxidation of NADH and FADH2 is occurred in electron transport chain. Okay, so if accumulation of NADH and FADH2 is taken place or if they are not oxidized, okay, so enzymes of Krebs cycle, enzymes of Krebs cycle are inhibited. Okay, and also ఇందాక మనం పైన చూసాం ATP, NADH ఇవన్నీ పోయి సిట్రేట్ సింథరేజ్ కానీ ఐసోసిట్రేట్ డిహైడ్రోజన్ కానీ ఆల్ఫా కెటోగ్లూటరేట్ డిహైడ్రోజన్ కానీ ఇవి వెళ్ళి ఇనిబిట్ చేస్తాయి. ఓకే, సో అంటే మనకు NADH, FADH2 ఉన్నాయి, చాలా ఉన్నాయి అంటే ATP ఆటోమేటిక్గా ఫామ్ అవుతుంది. ఓకే, సో ఏటీపీ సఫిషియెంట్గా ఉన్నప్పుడు ఈ NADH, FADH2 వెళ్ళి ఈ ఎంజైమ్స్ అవి బ్లాక్ చేస్తాయి. ఓకే, సో మనకు కనుక ADPs అనేటువంటివి రెడీమేడ్గా చాలా ఎక్కువ మొత్తంలో అవైలబుల్ ఉంటే సో ఈ NADH, FADH2 అనేవి ఆక్సిడైజ్ అవుతాయి. దేనివల్ల? ADP వల్ల. ఎక్కడ? ఎలక్ట్రాన్ ట్రాన్స్పోర్ట్ చైన్లో. అప్పుడు ATP అనేది ఫామ్ అవుతుంది. ఒకవేళ ఈ ADP లేదనుకుందాం, ఇది ఆక్సిడైజ్ కావడానికి ADP కావాలి లేదా GDP అన్న కావాలి అట్లీస్ట్. ఓకే, సో అవి లేవు అంటే ATP చాలా ఉంది అని అర్థం. సింప్లీ అది లేకపోతే దీస్ విల్ నాట్ బి ఆక్సిడైజ్డ్, దే విల్ బి అక్యుములేటెడ్ ఇన్ ద cell. Okay, so these will block these enzymes, so there is no Krebs cycle. Kabatti availability of ADP is very very important for the proceeding of the Krebs cycle. Okay, so yeah, ఇక్కడ ఏమవుతుంది, NAD+ and FAD+ కూడా కావాలి Krebs cycle కంటిన్యూ కావడానికి. Krebs cycle కంటిన్యూ కావాలంటే మనకు NAD+ and FAD+ కూడా కావాలి. ఓకే, ఇక్కడ ADP ఒకటి కావాలి ఎందుకంటే ఇవి ఆక్సిడైజ్ కావాలి. ఓకే, లేదా NAD+ and FAD+ అయినా రెడీగా ఉండాలి. ఓకే, సో అప్పుడే సో ద Krebs cycle అనేది కంటిన్యూ అవుతుంది. అండ్ ఇది ఒక పదం ఇది గుర్తుంచుకోండి, అడగవచ్చు అడగకపోవచ్చు, నాకు ఐడియా ఉండాలి ఆ పదం. ఇట్ ఈస్ యాంఫిబోలిక్ పాత్వే. What does it mean? It is both catabolic pathway and anabolic pathway. సంబంధం లేదు. So TCA cycle is యాంఫిబోలిక్ pathway. How it is catabolic? So here oxidation or degradation of, you can call it as pyruvate or acetyl acetyl CoA. Okay, metabolic, in that way, it is catabolic pathway. Okay, and why it is anabolic pathway? So here we have seen compounds like, alpha ketoglutarate, succinyl CoA. Okay, these are the intermediates of many, amino acids, the fatty acid metabolisms, in which many amino acids are synthesized. Many fatty acids are synthesized. So here, formation of alpha ketoglutarate and succinyl CoA, okay, they ultimately, if it is diverted, they ultimately lead to synthesis of some amino acids or some fats. Okay, ఆ రకంగా చూస్తే it is also, it is also an anabolic pathway. Okay, so అంటే చాలా other, so many amino acids and fatty acids needed the intermediates of this cycle. So in that way, it is also, the anabolic pathway. So here it is both catabolic and anabolic. That's why such kind of pathways are generally regarded as antibiotic pathway. So with this, we have completed the citric acid cycle, and but something is missing. Yeah, so two terms, the కొంచెం difficult to understand. So లేదు అనుకున్నాను. So here in aerobic cells, okay, so here there is a term called posture effect. So what is posture? It is a kind of, regulation of glycolysis actually. So inhibition of glycolysis by oxygen indirectly is called posture effect. So for example, there is ample amount of oxygen is there. Okay, so that leads to ultimate formation of the ATP and citrate. Okay, so if they are ample or sufficient amount of ATP and citric acid or other KREBS cycle compounds are formed. What does it mean? Only in the presence of oxygen, the ATP is formed. Let's take ATP. Okay, so in the presence of oxygen, the ATP are formed. So they are formed, more than enough ATPs. So these ATPs will go and inhibit phosphofructokinase enzyme, which is an allosteric enzyme. అంటే ఇక్కడ ఏది ఇన్హిబిట్ చేస్తుంది? ATP ఇన్హిబిట్ చేస్తుంది, లేదా సిట్రేట్ ఇన్హిబిట్ చేస్తుంది. దేన్ని? గ్లైకోలైటిక్ ఎంజైమ్ PFK ని ఇన్హిబిట్ చేస్తుంది. ఓకే, సో దీనికి కారణం ఎవరు అంటే ఇండైరెక్ట్ గా ఆక్సిజన్ ఉంటేనే ATP ఫామ్ అవుతుంది. సో దీన్నే పాస్చర్, లూయి పాస్చర్ ఐడెంటిఫై చేసిండు. ఓకే, సో డ్యూరింగ్ హిస్ రీసెర్చ్, దిస్ ఇస్ కాల్డ్ పాస్చర్ ఎఫెక్ట్. అండ్ Crabtree effect, it is anti-posture effect. Okay, inhibition of oxygen consumption by the addition of glucose to tissues having high aerobic glycolysis is known as Crabtree effect. ఇది అర్థం అవ్వచ్చు, కాకపోవచ్చు, రైట్? జస్ట్ ఆ పదం వినండి అంటే రైట్. సో ఏంటిది అంటే ఇక్కడ ఆక్సిజన్ కన్సంప్షన్ అనేది తగ్గిపోవడం. దీనివల్ల అంటే ఎక్కువగా గ్లైకాలసిస్ ఏ సెల్స్ లో జరుగుతుందో, ఏ టిష్యూస్ లో జరుగుతుందో, దానికి మనం అడిషనల్ గ్లూకోజ్ ఇస్తే ఓకే, ఆక్సిజన్ కన్సంప్షన్ అనేది తగ్గిపోవడము. దాన్నే మనము క్రాఫ్టరీ ఎఫెక్ట్ అంటారు అనేది కాన్సెప్ట్. సో ఎందుకు? డ్యూ టు ఇంక్రీజ్డ్ కాంపిటీషన్ ఆఫ్ గ్లైకాలసిస్ ఫర్ ఇనార్గానిక్ ఫాస్ఫేట్ అండ్ NAD+, which limits their availability for phosphorylation and oxidation. ఇక్కడ ఏమవుతుంది అంటే ఎక్కువ గ్లూకోజ్ ఇచ్చినప్పుడు గ్లూకోజ్ ఎక్కువ ఉంటే గ్లైకాలసిస్ అనేటువంటి ఎక్కువ జరగడానికి అవకాశం ఉంటుంది. అప్పుడు ఫాస్ఫేట్ కోసము NAD+ కోసము కాంపిటీషన్ అవుతుంది. ఓకే, సో కొన్ని ఫాస్ఫేట్లు కొన్ని గ్లైకాలసిస్ కి, కొన్ని NAD, కొన్ని గ్లైకాలసిస్ కి వెళ్ళి దే విల్ బి బిజీ విత్ glycolytic pathway only. Okay, so Krebs cycle ki sufficient amount of NAD+ are not available. దానివల్ల ఏమవుతుందంటే ATP అనేది అక్కడ ఫార్మేషన్ అనేది కుంటుపడిపోతుంది. ఇట్ ఈస్ హాల్టెడ్. ఎందుకంటే నెంబర్ ఆఫ్ గ్లైకోలైటిక్ పాత్వేస్ ఆర్ బీయింగ్ అక్కర్డ్ ఎట్ ద సేమ్ టైమ్ అండ్ దీస్ NAD+ అండ్ ఫాస్ఫేట్స్ ఆర్ బిజీ ఇన్ దట్ గ్లైకోలైటిక్ పాత్వే. సో there is no Krebs cycle. అంటే Krebs cycle చాలా ఉంది. ఈ గ్లైకోలైటిక్ పాత్వేసే సరైన NAD+ ఇనార్గానిక్ ఫాస్ఫేట్లు సఫిషియెంట్ గా లేక అవి స్లోగా జరుగుతుంటాయి. సో దెర్ ఇస్ నో Krebs cycle అండ్ దెర్ ఇస్ నో యూటిలైజేషన్ ఆఫ్ ఆక్సిజన్. సో దట్ ఈస్ ద మీనింగ్ ఆఫ్ క్రాఫ్టరీ ఎఫెక్ట్. సో ఆక్సిజన్ కన్సంప్షన్ అనేది ఇనిబిట్ చేయబడుతుంది. దేనివల్ల? అడిషన్ ఆఫ్ మోర్ గ్లూకోజ్ వల్ల. దానివల్ల ఎక్కువ గ్లైకాలసిస్ జరిగే అవకాశం ఉంది. ఎక్కువ raw material will be in competition to participate in glycolysis. Okay, that halts the Krebs cycle and electron transport. So that is called Crabtree effect. Okay, so this is glycolysis and Krebs cycle. So రేపు మనకు రెస్పిరేషన్ కంప్లీట్ అయిపోతుంది. రేపు నోట్స్ ఇస్తాను. అండ్ చాలా మంది అటెంప్ట్ చేయలేదు. ఎందుకో నాకు అర్థం కాలేదు. సుప్రియ ఇంకా అటెంప్ట్ చేయలేదు. సుమయ్య అండ్ సమత, రమ్య, అనిల్ కూడా చేయనప్పుడు, లక్ష్మి గారు అండ్ దిస్ ఇస్ మమత ఏదోనో చేశారు. ఓకే, గాయత్రి కూడా అటెంప్ట్ చేయలేదు. ప్రమీల, శ్రీదేవి గారు చేసినట్టున్నారు, రమేష్ చేసినట్టున్నారు. ఓకే, మీరు అందరూ ఆ టెస్ట్ అటెంప్ట్ చేయదమ్మా. నేను చాలా కష్టపడి రాత్రి 3 దాకా కూర్చుండి ఆ క్వశ్చన్స్ తయారు చేసినాను. అటెంప్ట్ చేయండి. ఓకే, ఒకవేళ ఫస్ట్ టైం తక్కువ వస్తుండొచ్చు, లేదా మీరు ఆల్రెడీ చదువుకుంటే ఫుల్ వస్తాయి. లేదు అంటే కొంచెం తక్కువ వస్తే రానియండి. సెకండ్ టైం అటెంప్ట్ చేయండి. దాని మీనింగ్ తెలుసుకోండి, మీకు తెలియని విషయం తెలుసుకోండి. కానీ టెస్ట్ పెట్టగానే మీరు అటెంప్ట్ చేయండి. ఓకే, రేపు కూడా మేబీ మార్నింగ్ ఉంటుందో 12కి ఉంటుందో ఐ డోంట్ నో. క్లాస్ సండే కదా, యాక్చువల్గా సండే రోజు నేను తీసుకోను. కానీ ఈరోజు 2 క్లాసులు అండ్ రేపు కూడా ఎందుకు తీసుకుంటున్నా అంటే నెక్స్ట్ వీక్ కొంచెం నేను ఒక 2 days రాకపోతుండొచ్చు. ఓకే, సో అందుకే ఈరోజు 2, ఇలా అయితే రేపు కూడా 2 తీసుకుంటాను. వరుసగా ఒక 2 days నేను వర్క్లో ఉంటాను. అందుకే వీలైనన్ని ఎక్కువ క్లాసులు తీసుకోవడానికి ఐఎమ్ ట్రైయింగ్. ఓకే, సో అది ఓకేనమ్మా. ఒక్క నిమిషం. Anil, have you attempted the test?
Sir, recording ఆఫ్ చేయండి.
చేస్తాను.